Welcome to week 2 of Anatomy and Physiology Lab. This week we're going to talk about histology. This week in lab we're going to have our first quiz, and just as a reminder, that's going to cover the material from lab 1 we covered last week, as well as the pre-lab content included in this video for lab 2. So be prepared for that. That'll be right at the start of our lab. Second thing we're going to cover is use of the microscopes. So there is actually a second pre-lab video discussing how to use the microscopes, how to care for the microscopes. And you can also refer to exercise 3 in the lab manual if you need some more information there. And then once we become proficient in the microscopes, we're going to move on to histology, which is lab exercise 6 in the lab manual. And of course, you have a lab workbook that's available on Blackboard, and you'll work through that, complete that during the lab meeting today. Well, the first question to cover today is, what is histology? So as you can see on the slide here, histology is the study of microscopic anatomy of cells and tissues. So for this lab, we'll be using the microscopes to look at these cells and tissues to see what they look like, how they're structured, and how to identify the different cells and tissues. Now, you should recall from lecture that tissues are going to be groups of cells that share both similar structure as well as similar function. And we're going to be looking at the four primary types today, starting with epithelial tissue, also connective tissue, nervous, and muscle tissues. And recall that each of those primary types is going to be further subdivided into multiple subtypes. So we'll have plenty to look at in the microscope today. The first type of tissue we're going to talk about is epithelial tissue. And this is the coatings, the coverings, the linings, basically the surfaces of the body and all the organs. So it's going to be coating pretty much everything in the body. And there are plenty of examples of this, like the skin and the mucous membranes protecting the body from the environment. There's also the linings of the blood vessels and of the hollow organs as well. And what you'll see looking through the microscope is there's a distinct apical surface of epithelial tissue. So it has this free edge. And that is not going to be touching other cells because it's going to be bordering the lumen of these organs. On the opposite side, the basal surface is going to be attaching to other tissues, like connective tissue, for example. So here in this slide, we'll see epithelial tissue bordering that lumen, which is that free space. So the apical surface is the edge that is not connected to any other cells. And then the basal surface at the base of the epithelial tissue is then going to be connected to the connective tissue underlying it. When we talk about epithelial tissue, we're going to want to discuss the naming convention. So there are several types, and they're going to be based on the number of layers of the cells as well as the shape of those cells. So the first part of the name is either going to be simple or stratified based on whether it's just a single layer of cells or whether it's multiple layers of cells. Add to that the general shape of those cells, and it gives you that full name. So simple squamous epithelium is going to be a single layer of these flattened, tapered cells. Cuboidal epithelial tissue is going to be more square-shaped, and columnar is going to be more rectangular. So each of these will have this two-part name. The first, either simple or stratified, depending on whether it's a single or multi-layered tissue. And the second is going to be the shape of those cells in particular. So that'll give us several varieties of epithelial tissue. And we actually have a couple more to discuss as well. These get a little more tricky because, as you can see, with pseudostratified, meaning false stratification, it looks like there are multiple layers of cells. You can see in the diagram over here that the nuclei are layered in different levels of those cells. But if you look closely, the cell membranes extend all the way from the basal surface out to the apical surface. So even though it looks like there might be multi-layers, there is actually only a single layer of epithelial tissue there. So those can be a bit tricky. You can see in this example that oftentimes these will have cilia, so that might help you in your identification, but be sure to pay attention to that in the lab today. The other one that can be tricky is transitional epithelium. And in this case, in the diagram, you can see there are multiple layers of epithelial cells, and you can see some of them are rather cuboidal near that apical surface there. Others are kind of funny shaped. And the key with this transitional epithelium is it actually allows that tissue to stretch. So you'll find this in the bladder in particular, where when the bladder is empty, you'll see it very well multi-layered. And as the bladder fills, it stretches out, it becomes to appear more single-layered. The second tissue type is connective tissue. And connective tissue is this structural and adhesive tissue, basically binds all the other tissues in the body together, gives them strength and gives them support. It's also the most abundant tissue type in the body, and you'll find it everywhere. And as you look through the microscope today in lab, you'll see that it's a very diverse tissue. So there are a lot of different subtypes, and based on their specific jobs, their specific locations, they can look very different, but they all can be distinguished by high levels of extracellular matrix. And this is that non-living, non-cellular material that fills in the spaces between the cells of the connective tissue. Now, connective tissue is going to be made up of cells as well as extracellular matrix. Now, the cells we see in connective tissue are going to be fibroblasts as well as lymphocytes. The fibroblasts live in the connective tissue. They're specifically part of that connective tissue. Lymphocytes are going to be white blood cells that are commonly found in connective tissue, but may be found in other places in the body as well. And then the extracellular matrix is that non-living, non-cellular material that fills in the spaces between the cells of the connective tissue. And the parts that we'll see is the ground substance as well as fibers. And the ground substance could be a number of things. It can be liquid, as is the case with blood plasma. It could be semi-solid, gelatinous, or even solid, as in the case with bone tissue. And we'll also find in there a number of fibers, either collagen, elastic, or reticular fibers, each of them having different strengths and different properties depending on the specific job of each of the subtype of connective tissue. Now, in general, what you'll find is those fibroblasts inhabiting the connective tissue there. They'll be generating extracellular matrix. You'll also see white blood cells like the macrophage here and the lymphocytes here that will also be found in that connective tissue. And then the three fibers shown here are collagen fibers, which are large, thick fibers, elastic fibers, which are a bit smaller, a bit narrower, and then the smallest are the reticular fibers. And then between all of those structures, you'll find this ground substance filling in the rest of the space. And when we talk about connective tissue, there's several different subtypes, as I mentioned. Connective tissue proper includes areolar, adipose, reticular, and denser fibrous connective tissue. We also include cartilage, bone, and blood as subtypes of connective tissue. Nervous tissue is the third tissue type we're going to talk about, and its primary job is going to be communication. And it has two distinguishing features, which are excitability, or this ability to respond to a stimuli, as well as conductivity, which is the ability to send electrical messages. In nervous tissue, we'll find two subdivisions. And interestingly, although cells are exceedingly small, neurons themselves may be up to 1 meter long. So these neurons are the specialized nervous cells that work to send and receive electrical signals. They're the most widely recognized, the most well-known cells in the nervous system. And you can see them pointed out here on the image of the microscope slide below. It's worth noting that the other subdivision is Neuroglia, which are the support cells of the nervous system, and they provide nourishment, support, and insulation for those neurons. Now, while these are not as widely heralded as the neurons, they're very abundant in nervous tissue and have an important job of protecting and supporting and nourishing those neurons. The final tissue type is muscle tissue, and this is going to be responsible for movement throughout the body. There's going to be three subtypes of muscle tissue we'll talk about, but each of these are going to be excitable, so they'll, like neurons, respond to a stimulus. They're also contractile, meaning that once they're excited, they will shorten. They have specialized proteins which will generate contractions and produce force. Now, the first subtype of muscle tissue is skeletal muscle tissue. And as the name implies, it's going to attach to the bones in the body, and it's going to be responsible for our body movements. Now, this is going to be voluntary muscle, so we consciously control its contraction. And if you take a look at the picture of the microscope slide of the skeletal muscle in the corner there, you can see it's very neatly arranged. It has very obvious patterns with striations or those lines running through the tissue. You'll also notice that you'll see a number of nuclei, and actually each muscle cell itself will have several nuclei in skeletal muscle. The second subtype of muscle tissue is cardiac muscle, and this is the involuntary muscle that's found in the heart. Now, this is only going to be found in the heart, so will not find it anywhere else in the body, and it is specifically set up to cause those contractions to generate pressure on the blood in the heart. What you can see from the picture here is that there's a lot of similarities to skeletal muscle. So you will see a somewhat parallel arrangement here, although there is a little bit of branching involved, so it's not quite as neatly arranged as skeletal muscle, but there are still the obvious striations running across that tissue. A couple important differences, though, is typically cardiac muscle will be uninucleated, so there is only one nucleus per cell. You can also see the structures called intercalated discs, which form junctions between those cardiac muscle cells. Now, the final subdivision of muscle tissue is going to be smooth muscle. This is, again, an involuntary muscle, so similar to cardiac in that respect, and it is found throughout the internal organs. So this is, again, going to be subconsciously controlled, and in this case, it actually looks quite a bit different from the other two subtypes of muscle tissue. So smooth muscle, as the name implies, does not have the striations. It has a smooth appearance to it. It also has these spindle-shaped cells, which are kind of flattened and short, so quite a bit different from what you'd see in cardiac or skeletal muscle. It's also, like cardiac muscle, going to have a single nucleus, so it is similar in that way, but it is fairly distinct from the other two types in some of those other aspects. As we wrap up with the pre-lab video here, a couple things to keep in mind for this week's lab is that, one, there is a whole lot of material, probably a lot of new material for you, and it tends to be fairly difficult for students. So we want to make sure you're making use of your resources. With your time in lab, make sure you're looking at the microscopes, looking at those different slides, being able to see and identify the different tissue types. Also recall you have those images in your lab manual. You can also find them online on a Google image search or a number of resources there. Make sure when you see those slides, you know what you're looking at, and you could tell me which subtype of tissue that is. And when you inevitably have questions, make use of your professor, make use of your TA in lab. They'll be able to help you work through the different slides and how you can identify all of these tissues. Because at the end of the day, you're going to have to recognize and identify these different tissue types. If you're shown a slide in a quiz or in an exam, you're going to have to be able to tell me what tissue type is it, what subtype is it, so make sure you're comfortable with that. And finally, take your time. 3 hours seems like a long time in lab, but it'll go by pretty quickly. There's going to be a lot to go over. So take a breather, take your time, make sure you're making use of, especially the microscopes and the slides, which you probably don't have at home. So you only have a limited amount of time to look at those. So that's one of your resources. Make sure you use that wisely, and we'll be in great shape for next week's quiz and the midterm in several weeks. Thank you.